Hello, everyone, and welcome to this Gen webinar titled From Models to Patients Harnessing Pre-Treated PDX Models for Precision Medicine. This webinar is made possible through sponsorship by Crown Bioscience. The poor translatability of early-stage preclinical models is one of the most significant barriers to oncology drug development. This is exacerbated by another significant barrier, interpatient tumor heterogeneity. Patient-derived xenografts, also known as PDXs, are the in vivo models that are considered to be the gold standard due to their demonstrated clinical relevance to human disease. In order to support the development of targeted treatment strategies and model resistance to standard and new therapeutics, it is necessary to unravel the biological processes that are driving tumor resistance to treatments, such as KRAS inhibitors and checkpoint inhibitors. There may be promising applications for pre-treated PDX models within the realm of precision medicine. These models are derived directly from the tumors of patients who were treated with known treatments prior to sample collection. During this Gen webinar, two industry professionals will talk about their experiences with pre-treated PDX models and share their insights. And a live Q&A session will follow the presentation, offering you a chance to pose questions to our expert panelists. My name is Jonathan Grinstein, Senior Editor at Genetic Engineering and Biotechnology News, a Marianne Liebert magazine that has been avidly covering the biotech industry for more than 40 years. Our guests today are Dr. Anthony Tolcher and Dr. Rekha Pal. Dr. Tolcher from Next Oncology will discuss the clinical histories of patients who have undergone multiple treatment modalities, including their significance and value in the process of developing translational PDX models. Dr. Pal from Crown Bioscience is going to talk about the process of creating pre-treated PDX models and the applications of those models in the process of developing and testing next-generation drug candidates. This is a reminder to stay tuned for the Q&A today after the presentation. All right, let's get started.
Thank you. My name is Dr. Anthony Tolcher. I'm Director of Clinical Research and also CEO of Next Oncology based here in San Antonio. And I'd like to discuss at least a little bit about the way and the view that I have for the future where PDX models will have to evolve. And the title of my talk is PDX models must continuously evolve, and that successful drug development results in new unmet need. Now, what do I mean by that? Well, we develop drugs based on the unmet need at the time. But with the success of new drugs being approved, suddenly we've moved the goalposts. And so the models that we used previously are no longer applicable. And I'm going to give several examples of this. It's really important to understand that with the Accelerated Breakthrough Program, we have so many new drugs being approved in the last few years that this has led to what I call development that seems to be chasing after what is the next best indication and what is going to be the next best unmet need. So if PDX models are going to be used to their fullest, they actually have to reflect the current clinical unmet needs. And as I said before, yesterday's models will not solve today's drug development problems. A perfect example is some of those breast cancer models that are ER positive and are resistant to hormone therapy. The whole field has changed. And in fact, the models that we should be using are those that are resistant to many of the newer therapies, the CDK4/6 inhibitors, as well as those that target patients who have PIK3CA mutations. So all of the models that we should be using as we try to move the field forward in a disease like ER positive or hormone responsive breast cancer need to be in those models because there is no value. There is no value developing drugs that predate the development and the success of the CDK4/6 inhibitors. So we really don't need a breast cancer model or a lung cancer model or a colon cancer model and so on, but rather we need a model that reflects the increasing fractionalization, you might say, of different indications. What we really need then is models that reflect how therapies have changed the disease. So it should be a breast cancer model that's HER2 positive, but is refractory now to trastuzumab deruxtecan. Now, that is important not only in the HER2 positive model space, but with the recent approval in HER2 positive low expressors, 1 plus to 3 plus, those are the models we need to use so that drugs that are being developed for that space after this agent, one can truly fulfill whether or not their drug is going to be useful. If there's no activity, for example, in models that are resistant to trastuzumab deruxtecan, then there's probably no point going forward with that indication. The same applies in lung cancer. If one has a non-small cell lung cancer model that has an EGFR mutant, it needs to be resistant to osimertinib. There's no point, again, in having a model that might be resistant to some of the older EGFR inhibitors. And we can say the same in lung cancer with the most recent approvals of adagrasib and sotorasib. Those tumors that have KRAS G12C mutations, they should be resistant to that. And in lung cancer that's HER2 positive, they should be resistant to Enhertu. Again, if we just look at another perfect example, there are lots of colon cancer models, but we really need to have colon cancer models that are refractory to adagrasib if they have a KRAS G12C. And so hence they have to be obtained from patients who have received this drug on top of, say, cetuximab or an EGFR therapy. Or for those that have the BRAF V600E, they should be resistant to cetuximab and encorafenib. So again, the models have to reflect the current unmet need. Increasingly, we're going to need models that are also resistant, you might say, to some of the newer approved therapies. So sacituzumab govitecan, this is an important drug in triple negative breast cancer and bladder cancer, and perhaps in lung cancer as well. So this is starting to fill those spaces. So newer models should have at least come from patients who've received this drug. With the recent approval of mirvetuximab soravtansine, we probably need to be thinking about ovarian cancer models that are both folate receptor alpha positive and refractory to mirvetuximab soravtansine. We also need models that are fully characterized to understand why and why not these new agents might work or might not work, quite frankly. So it's an important aspect here. Having models that might be resistant to the newest therapies is one thing. But if you're coming along with an agent that you think will convert a resistant model into a sensitive model, you need the full characterization. Was it because the target changed or diminished, say HER2 or KRAS? It's not unknown that certain types of KRAS mutants may become the predominant molecular genetic alteration, leading to resistance of the newer KRAS therapies. The same applies to HER2. Is it actually a loss of HER2 expression leading to resistance, or are there other mechanisms of resistance, such as trafficking or changes in efflux pumps? So that's why these models need to be fully molecular sequenced, and we need also RNA expression as well as proteomics. Otherwise, you could be seeing your drug not work and not have an understanding as to why they may not be working in these particular PDX models. Now, I'm a clinician, so I have a very different point of view of drug development than, say, some people who are laboratory-based researchers or drug developers. What we don't need, and what is not innovation, is coming to the clinic with another KRAS G12C inhibitor. There are more than 12 in clinical development worldwide, many from Asia. And it is not going to be of any great value to have a drug that targets this when we already have 2 approved, unless it can demonstrate that it can convert a resistant patient into now a newly sensitive patient. So if one comes along and says, we have a KRAS G12C inhibitor that works in models that are sotorasib and adagrasib refractory, and those patients, then that is something that is innovative and should be explored. Now, again, it is not it's not coming up with an agent that's following the herd. It's coming up with newer and better agents because that is the population we need new therapies for. Capital is tight, and many of the biotech venture funds, as well as private equity, look at biotech a bit differently than they did maybe 3 or 4 years ago. Everyone says capital is tight right now. So if capital is tight, what's going to be important? Differentiation. So you need to approach this as saying, why is your drug going to be different than the many others that are in the same space and still competing for the same space? So differentiation is going to be key. And I always say that, some of my interactions with these people in venture capital, they've seen it all. They have certainly seen all the different agents as people are scrambling around trying to get investments. And so they tend to know what is actually looking like it's going to differentiate themselves versus those that are me too, or attempting to be, me better. They have many companies to choose from, and that's important. So an argument to get funding right now is to say our drug meets a new unmet need in the accelerated approval or breakthrough designation because our drug works in models that are resistant to the current newest standard therapy. We're in this golden era of clinical cancer trials. In fact, this article from Nature Medicine in April of 2022 is important because it demonstrates the fact that we have never had it so good in drug development. And so there's a lot of different trials, a lot of different agents. And we are getting to the point where I would say there's almost more trials than there are adequate numbers of patients in the usual places where we have patients go on clinical trials. So we have to stand out, and the drugs have to be innovative. They can't be essentially lookalike drugs. And therefore, one needs to develop therapies that are to the patients that are resistant right now to the newest therapies. I like to say anticipate the future because it's coming regardless. And so what are the new agents that are coming along? Well, for people like myself, I expect to see an MDM2 inhibitor approved in the next 1 to 2 years in liposarcoma. So one needs to start thinking about the fact that you need to have models that might be resistant to MDM2 inhibitors. We'll see many more KRAS inhibitors in the next 3 to 5 years. Some of the KRAS on inhibitors, for example, that will cover the G12 mutations. WEE1 looks promising in serous uterine cancer, whether one drug gets approved or not, but one should start to plan that these drugs may very well be approved. And so you're going to be looking for therapies that are after WEE1. And of course, there are some drugs that have made some progress, and might have some promise in CEACAM5 lung cancer. And so lung cancer may change again, and we need to have therapies that are covering these particular agents. So one should anticipate that these drugs may or may not be approved, but they may be approved. And so there is no point coming in with a drug that doesn't somehow work in some of the patients who have been on clinical trials or received these drugs when they're newly approved. We've all seen the reports that menin inhibitors seem to be important. There may be resistance to that. Again, it's coming. And so one needs to start thinking, can your drug work when some of these other agents don't work? MTAP deleted patients, important population, very well-defined patient population. We've got a number of interesting and exciting agents targeting PRMT5 as well as MAT2A. One needs to start to think, okay, my drug will probably come after these current crop of new agents. So what is the agent I'm going to be developing? And then we've seen time and time again, immune resistance is a problem, an unsolved problem, you might say, because we can't necessarily predict what the mechanism of resistance to currently available immune therapies. And so we've got to start looking at these models and see if we can find some sort of approach, new immune therapies to sort of reactivate the expression of these neoantigens. So in summary, models, especially PDX models, must continuously evolve, and they need to reflect the current unmet needs, not the unmet needs of 4 or 5 years ago. The models need to be fully characterized because otherwise they will fail. You will develop a drug, but you may not know or understand why your drug works or why it has failed in these model systems. One really has to say, go for innovation, not the herd. I kind of get a little dismayed by the fact that so many drugs are being developed that really do nothing more than reflect the same targets and just say that their drug is going to be better, but they don't have the proof, at least in preclinical models, that they are better. And differentiation is going to be key. And one should anticipate the future. One can easily look at some of the most promising agents that are presented at meetings worldwide, and you will know that those drugs will likely be approved, and that may very well impact your development strategy. So trying to have models that reflect those future drugs. So with that, I'd like to thank you for your attention, and I'll pass this on.
Hi, my name is Rekha Pal. I'm a Senior Director of Scientific Engagement at Crown Bio. I want to thank Gen Webinar for giving us the opportunity to present Crown Bio PDX model platform here. And today the topic of my talk is establishment of next generation pre-treated PDX model, a tool for precision medicine. Today's talk is going to cover the three aspects as listed here. The first one is why do we need a clinical relevant PDX model? How we are establishing the pre-treated PDX model, and how we are doing the validation at Crown Bio for pre-treated PDX models. So why do we need PDX models? So PDX model is the well-known, very closely translation oncology preclinical model which bridged the gap between lab and clinic. And Crown Bioscience offers a panel of validated PDX model around more than 2,500, which include the different cancer type, and all these PDX model are very well characterized for in vivo preclinical research studies, as well as in vitro organoid development. Today, my topics mainly focus on the PDX model. So PDX model better represent the clinical heterogeneity of the patient compared to cell line. And the main features are it preserves original tumor histo and molecular pathology. It recapitulates intra and interpatient heterogeneity and patient diversity. And this is one of the most closely translation oncology preclinical model for pharmacologically predictive models data for the drug discovery program. So as I mentioned before, that the PDX model are gold standard preclinical in vivo model that reflect the clinical heterogeneity and diversity. And PDX model need to involve today unmet clinical need and model resistant to the standard and new therapeutics. We need these next generation PDX model so that the researcher and drug developer can access to these most clinically relevant model that most accurately reflect the patient population and cancer treatment that are currently being used. So these are the few platforms which we offer at Crown Bio. You can see we have three platforms here. The HUE trial is basically a mouse clinical trial platform in a preclinical settings. We can design this HUE trial based on, the access of the more than 2,500 PDX in vivo tumor model. And because these PDX model has already been validated for full RNA-seq as well as WES data, which can be useful to discover potential biomarker for the response and non-response. We can also go for the HUE mark. HUE mark is basically because all these PDX model has full RNA-seq and WES data and has been comprehensive annotation of these models to enable targeted drug development. And then we can also go for the discovery of the relevant genetic signature or pathways because of the RNA-seq and WES data available from the PDX model based on the drug responder or non-responder to the PDX model treatment. So this is the three study design which we have used for the mouse clinical trial. The first study design left-hand side is the 1+1 study design where we can have PDX model from multiple indications and multiple target. We can have one mice in each group, and what it will do, it enhance the robustness through the vehicle treatment animal. We can also conduct large studies, basically a screening kind of studies across multiple tumor type with limited animal numbers. The second study design could be a one type of cancer with different genetic mutation. This helps us to discover the biomarker within one disease, and it also accesses the target dependence in the case of target negative or low in specific PDX model. And the last study design is the multiple type of cancer with one common genetic mutation, such as KRAS or BRAF, right? So we can go with this study design, help us to provide robust target validation if your drug is targeting a specific mutation or a specific, a specific targeted gene expression. It can also explore the resistance mechanisms in the case of target positive and yet not responding to the specific treatment. So this is the Crown Bio online PDX database, which we call it HuBase. So this includes 30 cancer type. This has more than 2,500 PDX model. This database also include for each PDX model, the full genotypic and phenotypic data, which include the patient information, growth curves, tumor growth kinetics, any shareable SOC treatment data, full RNA-seq invest data, WGS data, and we are slowly adding proteomics data. So at present, we have around 139 PDX model with the proteomics data, but in future, we would be able to add for all the PDX model proteomics data. All these genomic as well as proteomics data can be searchable across cancer type for the gene expression, copy number, microRNA expression, mutation, or gene fusion. And we have recently added the pre-treatment filter where you can select the PDX model with the full clinical treatment history information. So now I'm going to talk about the pre-treated PDX model characterization. So this is a slide where we are showing the cancer type included in the pre-treated PDX model. So you can see we have added, major cancer type based on the patient treatment history. So prostate cancer, pancreatic cancer, gastric cancer, breast cancer, lung cancer, and colorectal cancer. And the right-hand graph shows the patient treatment history in relation to the immunotherapy, immune checkpoint inhibitor, hormone therapy, and targeted therapy or combination therapy. So these are the unique PDX model derived from the patient biopsy sample, which has the full treatment history, mainly involves, the current therapeutics such as immunotherapy and immune checkpoint inhibitor. This is the slide to show the pre-treated prostate cancer PDX model we have added. So you can see these are the 8 PDX prostate PDX model, which include both CRPC, which is castrate-resistant prostate cancer, as well as castrate-sensitive prostate cancer. These PDX model has the full patient treatment history, as you can see, for hormone therapy and any other chemotherapy treatment response, and also been validated and for the specific mutation or fusion pattern of the specific gene. All these prostate PDX model also has been validated for the immunohistochemistry for the prostate specific marker such as PSA and AR expression. Now, this slide shows the pre-treated model of the drug resistance. So mainly these PDX model, which include the majority of the colorectal cancer type, these PDX model has been generated from the patient treatment biopsy sample with the different treatment regimens, but mainly treatment by the KRAS inhibitor. So you can see the comment section, we have the KRAS mutation data, as well as the patient treatment response with the KRAS inhibitor, whether the patient was responsive to the KRAS inhibitor or not. And this is the very unique slide where we can see that we have added some of the PDX model, which from the patient biopsy sample, which were treated with the immune checkpoint inhibitor. So those are kind of current therapeutics treatment regimen going on, with the mainly focus on the immune checkpoint inhibitors. And these are unique PDX model, which include various cancer types such as lung, as well as the colorectal cancer and the endometrial cancer. And these patients were resistant to pembrolizumab or nivolumab, as you can see in the comment section. So now I'm going to show some of the case studies, what we have done for the establishment and validation of these pre-treated PDX model establishment. So this is CR9537 colorectal PDX model, which has been generated from the patient biopsy sample, which has been gone through a various treatment regimen and also been treated with the KRAS G12C inhibitor. The patient was responder in the beginning but became resistant. We have validated this model in the BALB/c nude mice, and then we also have done the PDX efficacy response data by using the 2 KRAS inhibitor Amgen 510 as well as MRTX 849. And you can clearly see from this graph that the CR9537 PDX model was not responsive to the MRTX 849 treatment, and as well as the partial response to the AMG 510 treatment. This is another colorectal cancer PDX model 9547, which has also been derived from the patient biopsy sample, which has been treated with the KRAS G12C inhibitor. The patient was responder in the beginning and then become resistant. We have validated this model in BALB/c nude mice, and this is the tumor efficacy data in this PDX model for the 2 KRAS inhibitor Amgen 510 as well as MRTX 849. And you can clearly see this model was resistant to the MRTX 849 treatment, as well as partial responder to the Amgen 510 treatment. So this is kind of validation that what the patient was showing in the clinic after the PDX model establishment from the same patient, we are observing the same treatment response and phenotypic data. This is one of the prostate cancer 6511, which was a castration resistant, and we have validated this model with the anti-androgen treatment. And you can see with the both treatment, enzalutamide and abiraterone, this PDX model was not responsive to the treatment. This is one of the breast cancer 9456 PDX model characterization. This patient was positive for lung metastasis and received paclitaxel treatment before establishment of PDX model from primary site. This one also majorly mutation in BRCA2, TP53, and DNMT1. We have validated this model for tumor growth kinetics in both NOD/SCID and BALB/c nude mice, and then we have used the tumor chunk for the IHC staining for ER negative, PR negative, and HER2 positive. Further on, we also validate this model for the SOC data, so you can clearly see from the right-hand graph for the tumor efficacy data, where this model was responsive to lapatinib, was responsive to capecitabine, but was limited response to lapatinib, tucatinib, and paclitaxel. And this model was also responsive to the combination of tucatinib and capistatin. So this is my last slide for the conclusion, that these PDX model are the gold standard in vivo model for their proven clinical relevance to human disease, because it maintained the primary patient sample heterogeneity, as well as closely recapitulating the predictive value of the drug treatment response. This next generation PDX model is very important for unraveling the biological process, which might be responsible for tumor resistance to the treatment. And that's why we have included the KRAS inhibitor and checkpoint inhibitor, which are necessary to develop the targeted treatment strategies and model resistant to standard and new therapeutics. The current preclinical model and the drug discovery program needs these pre-treated PDX model, which directly derive from the tumor of patient who received known treatment before sample collection, and to have promising application for precision medicine, as well as the establishment of the next generation drug therapeutics. So thank you so much for your time.
Hi, everyone, and welcome to the live Q&A portion of the webinar. Please make sure to submit any questions you may have for the panelists now in the chat box. My name is Jonathan Grinstein. I'm Senior Editor at Genetic Engineering and Biotechnology News. I wanted to thank Dr. Anthony Tolcher from Next Oncology and Dr. Rekha Pal from Crown Bioscience for those wonderful presentations on pre-treated PDX models. And again, this is the live Q&A portion of the webinar, so please submit any questions so we can ask them to the panelists. To kick off the Q&A session, here's a question, I think pertaining to Dr. Pal at Crown Bioscience. How many pre-treated PDX models does Crown have?
So we have around 338 pre-treated PDX model. And if you go to the online Crown Bio Hubase database, you can select, there is a tab, as I mentioned in my talk. So you can select the pre-treated tab and look for all the pre-treated PDX model available.
Is there some sort of like groupings or kind of general categories there that can be found?
Yeah, so it includes various solid tumor types. You can select. Yeah, so grouping means we can include, you can see all the cancer types. I don't know, I'm hearing echo here a lot. So it's kind of, maybe it's just happening.
Okay, I'm not sure if we're having some technical difficulties there, but let's move on to the next question. Let's see. Here's a question from the audience, I believe. Complete omics characterization of a PDX model can be expensive. How can you overcome this expense in small research settings, such as in small companies?
Well, I'll jump in. I'll move it over to Dr. Paul. But one of the big ways to make it successful is that with a large database that's actually already been characterized, then you're actually only paying a fraction of the cost, because that model is then being used and leveraged across multiple different customers. It's always difficult if you say, okay, well, we want to have full genomics on those patients and you're paying for the entire amount. That can be a bit problematic. So sometimes the larger companies have already got that wonderful database. And so in the end, you're able to leverage that size and that work that's already been done.
Yeah, can you repeat the question? Now I'm okay. Yeah.
Sure. It's complete omics characterization of a PDX model can be expensive. How can this be overcome, or how can this expense be overcome in a small research setting, such as with small companies?
So yeah, so this is why we are characterizing full. It's really expensive to validate all these models. We are adding next generation PDX model with the full clinical treatment history. So that's the first step we are doing at Crown Bio, providing all the RNA-seq and West data. In fact, we are also using some of the SOC to make sure that, as I was showing in my presentation, that the patient was not responsive to the specific treatment. We should see also in PDX model once we validate it. So we are doing all the first step. In fact, 50% of work are all very well characterized PDX model to offer you as a service, to go right directly into diving into the drug discovery program. So please feel free to go to our database, check out the model based on whether you have any drug target specific selection or any mutation specific selection. We do have the drug tab also, because we do have a lot of in-house, our own R&D data. So you can select specific drug also if you are in your pipeline, if you are developing a therapeutic, which might be, looking for your combination with the specific SOC or any other targeted therapy. So you can see we have humongous amount of data already sitting there, which will help you to actually basically just go and look for, the model selection and then design for your study design based on your drug therapeutics target or any other criteria you have.
Great. Here on to the next question. Does Crown Bio have some standard of care treatment response data in pre-treated PDX models to compare to patient treatment responses?
Yes, so that's as I mentioned before, and I showed in my slide also, we are getting this kind of question when I'm talking to the investigator, drug discovery researcher, that they want to see the validation also, because there are so many times the parental, sample does change the phenotype, and which is very critical. Like we get the sample grade, but do we able to maintain the same heterogeneity? Do we able to maintain the same genotype or phenotype, and also the drug treatment response. So we have right now some of the models already been validated for the SOC also for the same drug, which was treated for the patient too. And we are slowly adding. We know the importance of to maintain the phenotype, especially in the drug treatment and response and the genomic integrity and heterogeneity of the tumor. So hopefully in future, we will slowly adding all those drugs which has already been in clinical, in clinic has been treated for the patient, and then we got the sample. So that's our kind of pipeline going on. And that's why I've shown some of the case studies where we are seeing very clear pattern of maintaining the same treatment response as patient was in the clinic for the drug.
Here's a question, I think, for both of our panelists. What are your thoughts on using these pre-treated and other PDX models to evaluate IO agents in nude mice that lack T cells?
So yeah, that definitely we are not going to. That's why we have no IO agent treatment response. So that's we will go jump onto the humanized system, right? So we will go with MSC1 and 2, which is engrafted with just recently been launched. We are doing some of the studies, which is kind of cost effective compared to CD34 engrafted mice. We are running awesome studies for the screening of especially PDX model. So we have planned to do that for immune checkpoint inhibitor to check these PDX model for the specific immune checkpoint inhibitor treatment response using the humanized model system. And I think MSC1 and 2 knockout engrafted with PBMC or the CD35 engrafted mice would allow us to do that.
What about PDX humanized models for T cell engagers evaluation with various pretreatments, including ICI? Do you have any thoughts on that?
Yeah, so so T cell engager, you can select these models if we have already patient treatment information for immune checkpoint inhibitor. We have response data also, not for all of them, but some of them definitely. So those can be used for for CD35 engrafted, or if you're looking for MSC1 and 2 PBMC engrafted mice. So yes, these PDX model are ready to validate, already been validated for tumor growth kinetics. So we can jump onto the humanized system to look for T cell engager treatment response data.
Here's a question for Dr. Tolcher. As a collaborator here, how do you go about obtaining your specimens that you from working with Crown? And can you tell us a little bit more about like the interaction there?
Yeah, so for us, we're a phase 1 center. So patients come to us and are referred to us to go on clinical trials. And frequently they have mandated biopsies that are performed. And so we obtain the specimen at that time. And if you think about it, anytime a patient is coming for a clinical trial, we then document all the prior therapies and usually the prior response that's necessary. And so we're able to capture the clinical history of the patient as well as the responsiveness and the amount of prior therapy that they've had. At which point, if there is some leftover tissue in a biopsy, we implant them and grow them up. And then if successful, that's how we collaborate with Crown. We make all of our PDX models available to Crown in a wonderful working relationship. And that really does allow then those models to be expanded and used for others to further drug development.
There's a few questions here kind of going back to the first question that we kicked this all off on, which has to do a bit with the Crown Bio database. There's a question on, can the model database, can you filter pre-treatment PDX models using the database itself to kind of, hone in on specific pre-treated PDX models?
Yes. So if you can contact us, we can review, I can show you by myself how to do that. Or there is a left-hand side, there is a tab for pre-treatment. Once you open to the HuBase, so just click that tab select that model. So you will come up, you will see all the 338 pre-treated PDX models. So you can do all, based on your spec. Mostly they are all the solid tumor type. So select the model based on the, yes, so we do have tab left-hand side. You can see the different tab for it.
And I just want to finish or come back to that very first question, which I think we had some trouble with technical difficulties. How many pre-treated PDX models does Crown Bioscience have?
Around 338, and we are in the process of adding more.
Great. Kind of going back to like the science side of things here with the question, back to what Dr. Tolcher was talking about. Is there a concern of using a single PDX tumor to evaluate a drug response, or is it recommended to use multiple patients to ensure you're not seeing a patient-specific response?
Yeah, no, I think actually, like anyone, we should be diverse in terms of the number of tumor models one explores, because you don't want to be traveling down an idiosyncratic situation. So, I think using sort of the N of one kind of studies where you sample for a number of different models and then expand and go into more depth is sensible. It also provides you with, I think, some clues. If your drug works or doesn't work, in one particular model, it's probably useful to at least understand why. And by looking at the responders and the non-responders, that information could be incredibly valuable in terms of biomarker selection, because not all drug is going to work in every patient with a given indication. And to improve your odds when you're in the clinic of finding and identifying those patients who have responsive disease, it really does probably require quite a large selection of responders and non-responders, so that you have some confidence that you're pursuing a sensible drug development path.
Here's a question to Dr. Pal that I think stays on the same subject, but you mentioned that using an N of 1 mouse in the basket cancer screening HUE trial to initially test compounds and understand efficacy. How do you reconcile the robustness of a non-response with just one mouse?
Yeah, if you ask me, I always recommend to go with N is equal to 3, but we do have published paper with N is equal to 1. So say you selected 20 PDX model for the HUE trial, right? And if I'm as an investigator, I would like to see each model response too. But in HUE trial, what we are doing exactly putting all the control group in one basket and all the treatment group in one basket. Just like in clinical trial, right? Every patient is different, even if they have the same cancer type, same grade, they differ, because their genetic information is different. So we are also going to see the same. So I always recommend to go with N is equal to 3, so which give me for each model response data to compare to the control group. We do have, this is where we do the t-test analysis, putting all the control in one basket, and then all the treatment group in other basket, and look for the, significant value. But 3, N is equal to 3 would be the grade to add. So N is equal to 3 for each group, for each PDX model. And if you're interested, we can send that paper, please contact the Crown Bio or me, and we can send that published paper to show how we have some of the case studies also using the HUE trial, as well as the published paper.
Here's a question I think that, in general can be directed towards Crown, but I think it'd be interesting to get Dr. Tolcher's take on it, which is where are the PDXs implanted? Is it the same across all the mice that you have at Crown? And is there some work there that can be done to create more specific models, or choosing where the implantation of PDXs is?
Yeah, if I'm interpreting the question, so that, it's in the flank, of course, subcutaneous. It's not, we don't do orthotopic here. Once the model has grown to a certain degree, then we transfer it over to Crown to use their expertise to expand it. But we are not a group that does orthotopics. If somebody is thinking about that, I think there's some benefits, but there's also some disadvantages about orthotopics and the complexity. And does a orthotopic really reflect much better than a PDX? I think the jury is out on that. Certainly, it adds to complexity.
This one's for Crown and Dr. Pal. Are all your mice, or are your mice humanized? And if so, how are they being humanized? Are they humanized with human CD34 or matching PBMCs? And kind of going further into the question, do you have genetic characterization of your PDX passages? Since they tend to get aggressive, and what passage do you usually use for the drug treatment? Several questions in there.
Yeah, so I will go one by one. So first one is that, as I shown in my presentation, we first use it as a xenograft. So it's immunodeficient mice. Either we are using Babcineud or Northskid. If you are looking for any kind of, bispecific or immune checkpoint inhibitor treatment response, a drug which is kind of reacting with the immune cell population, we need to go with the immunized. We are using PBMC engrafted based on the tumor growth kinetics of the PDX model. If the PDX model is, because PBMC engrafted mice, we have very short window, 3 to 4 weeks, unless the GVHD issues comes up. And that's why we don't recommend to go with PBMC engrafted mice for the PDX model immunized study. We usually go with CD34 engrafted mice, which has the no GVHD issue. And recently, as I mentioned before, MHC1 and 2 knockout mice, we are doing studies with the PBMC engrafted. It gives us around 70 to 90 days, so which is enough for the PDX model to see the drug treatment or response. Usually PDX model randomization criteria, once the tumor volume reach 80 to 120 millimeter cube, is around 20 to 25 days average, except the prostate or some other model which are really slow grower. And then you can start the treatment. And this second question was or like genetically, right? So how the different passage number, we try to go P11 maximum, but there are some of the PDX model which is high demand, which a lot of other investigators using it. And we went high, we are doing study with P13 or P20 also. But in that case, we always do QC. So whenever we passage, we do H&E staining. That's kind of first step, QC, to look for any kind of morphology change. We have the clinical grade pathologist. If we are seeing any slightly different in heterogeneity or the morphology of the tumor, we go for again RNA-seq canvas data to make sure that it matches, the parental sample with the genomic information. And that's what we are creating organoid model actually from the PDX model. So we call it PDX. So I didn't bring it up because this webinar was focused on PDX, but we do have organoid generated from PDX models. So it's kind of in vitro system where we are doing again genotyping, means WES or RNA-seq data, genomic data for using the organoid model itself, and then comparing with the PDX model how much concordance between the mutation and expression pattern, and we are seeing almost 97% concordance. So we are very careful doing that, but we do have for some of the model multiple genomic information for different passage number if we have seen slightly difference in heterogeneity of the by H&E staining. So these are kind of our routine rigid QC, for PDX model.
Here's a two-part question from the audience. I think the first one maybe can go to Dr. Tolcher and then follow up by Dr. Pal. But how important is IO response data in selecting a model to study in combination therapy research? And the second part of it is, how many Crown models have full response data from currently approved checkpoint inhibitors?
Yeah, I think actually having IO responsiveness in certain malignancies, 17 plus indications that IO is approved for. And I think that, from a, just a clinical standpoint, that is the reality. It's moving closer and closer to frontline therapy. So for patients and samples that come from patients who've not experienced IO and you don't have the information on, to me, you're leaving out one very important aspect of their prior therapy. Now, can those be used in specific, IO, PDX models. That's not my expertise, and I'll leave that to Crown because they have model systems that they try to explore in that circumstance. But I think it's really important that we encompass the entire standard of care options for those patients and in those models that we're obtaining, so that you're actually working with what I call the true clinical situation on the ground that you would be treating in the clinical trial.
And so the second part of the question for the Crown, we do have most of our humanized study from the client studies, because these are very expensive studies, and we cannot validate all the model for the humanized. So, and there is no validation because it's always donor to donor variability. So that's why we validate the tumor growth kinetics, for the model first in immunodeficient, and any model which is validated can easily go to the humanized system, because we have tumor growth kinetics, full genomic data, as well as a lot of treatment response. So we have around 50 PDX model for the immune checkpoint inhibitor. Mainly, majority of these data are from the client study, bispecific, and and we do have a lot of PDX models. Some of the PMAs, like quite good amount of PDX model with the CAR T therapy also. So yes.
Here's a follow-up question from the audience on the passaging information. Do you at Crown Bioscience have genetic information of each passage of the PDX?
It's impossible to do each passage of the PDX for the full genomic. Means we are already providing so much, and that's why we are very, we are very aware and very careful. We have a rigid QC. That's our HNE. It does not need to do the RNA-seq for each passage number unless you see some differences. And HNE staining with the pathology grade clinical pathologist will tell you if there is any drift in the morphology. And that's why if you access our database, you will see some of the models. We do have multiple passage RNA-seq information, not a lot of model, but some of the model where we have seen. And I forgot to mention before also that we do have FFP slides also. For all the PDX model, we do have TMAs also. So if you are looking for any kind of specific target using our gene expression, by genomic information, you can always get these slides, which are FFP slides, and use for IHC for the specific target validation. Even you can use the same passage number, which we are going to plan. We will let you know, you decide this passage number is going to use for your study. So we can use the same passage number for the IHC staining of your target. It could be 5 or 6 target. And that's what the reason we are adding proteomics data also. So look at how much Crown is investing, to fully integrate the full information of the PDX model, because there are cases where we have seen differences between the gene expression and the protein expression for the same target. And that's what the reason we are slowly adding the proteomics, but you are most welcome to get the FFP slides. We can do it for you for the IHC for your target, or we can send it to you and you can do in your lab facility to look for the target or specific expression in the specific PDX model.
Here are 2 audience questions grouped together because they kind of have to do with the database. One is, I noticed the list of PDX models by patient treatment history doesn't include ADCs. Is this under the treatment tab, perhaps unspecified, or is it missing? And then the second one is, are there any treatment-resistant syngeneic models available, such as anti-VEGF resistant lines?
Yeah. So okay, first question is for PDX. Second, I will also answer for the syngeneic. ADC, we have majority of data from the client study, so that's why we won't be able to put it on. We don't share client data, but if you are looking for any specific ADC treatment response, please do let us know. We might be running in-house. We are planning to add some of the data in the data based on our own R&D data, as well as, we can share in Excel file about like how much experience we have with the ADCs. The second question is for syngeneic. If you go to the Mu base, if you're accessing database, please go to the Mu base. you will see, just select the model. You will see the syngeneic model. Just select that tab and you can see a lot of treatment response for the specific, or, please contact us or contact me. My email is rekha.pal@crownbio.com. So I'm going to send you all the data packs and any questions you have for relation to the syngeneic or the PDX model.
And one more specific question along those lines. Do you have PADCEV pretreated urothelial carcinoma PDX models?
Urothelial, I need to double check, but I don't think we have any clinical information fully urothelial, but we do have PDX model. But I will double check with the team and then let you know. Yeah, please contact me. I already put my email here. So yeah, please feel free to contact us anytime.
Here's an audience question that I think is kind of more conceptual, if you will. Is there a graphic or a measure that would show the beginning of cancer until survival of cancer until survival cancer for a period of 2 to 3 years? So this person says that I'm mentioning this because there are laboratory analyses before and after chemotherapies that would give direction also after the survival period, that are still certain indicators that the human body still saves as memory for that cancer. I'm not sure if that came across. Yeah, so I guess I think the question is trying to say, are there any methods that you use to test, like if the immune system has created memory for the cancers that you test, or any other measures beyond just survival, I suppose?
Yeah, actually, this question comes up recently with one of my discussion with some of the drug discovery investigators. These are the PDX model. We are getting biopsy sample. We are inoculating in the immunodeficient mice. So there won't be, if there is any immune cell other population, it won't be survived too long. So basically, it's a very pure tumor cells population in the PDX model. So we have no characterization of the memory cell coming from the patient. Maybe we can see this phenotype. We have this EVPT program. It's called ex vivo patient platform, where we are using fresh patient sample coming with the biopsy sample intact. Sometime, not all the time. Immune cell population. So we can do 30% of the sample go for full characterization and then go for co-culture system. But this is going to be all in vitro system. We do get sometime blood also from the same patient. So if you're interested in EVPT, please contact us. And I just looked up in the database for before this question, like, do you have uterine cancer for any pre-treatment? So we have 4 uterine cancer PDX models. So yeah, please check it out. Check the pre-treatment tab. So it will give you there's a 4 PDX model there.
And one last question here we have from the audience, one that I think Dr. Tolcher would appreciate and maybe be able to comment on as well. But engrafting PDX can be a huge pain in terms of its success rate. What are some of the learnings that you have uncovered to help increase success rates of engrafting patient-derived tumors into nude mice?
Yeah, so, I don't do the engrafting. I have a team that have expertise in doing that. Many of them have, 10 years experience doing this engraftment. And so there are some tissues that will naturally engraft much better. So certain colorectal, pancreatic, ovarian, for example, those will engraft very well. We find that pleural fluid tends to engraft exceptionally well. Others are more difficult. And there's a good reason why so few PDX CROs have prostate cancer models, because those are very difficult to get to grow. So it's really experience on the staff, and then also the particular indication, and those factors really play a large part into the success of engraftment.
Okay. And with that, we've come to the end of our webinar. I'd like to remind everyone that this webinar will be archived on the GEN website, which is at genengnews.com. We'll be there for up to a year. So if you missed any parts of it, you can watch it again, or feel free to forward it to any of your friends and colleagues who you think might be interested in the content or the webinar itself. I'd like to thank Dr. Tolcher and Dr. Powell again for this incredibly informative presentation. And I'd like to thank you, the audience, for your attention and some really great and thoughtful questions. Very special thanks goes to Crown Bioscience for sponsoring this webinar. And hopefully we'll get to see you again at another Gen webinar in the near future. Goodbye for now.